Welcome to your next video notes for Unit 6. Last class, you learned about protein synthesis and how DNA can be used to make proteins. This class, we're going to learn about errors that might happen in that process of protein synthesis and how that leads to mutations. You might have heard of the word mutation before. A mutation is a change in the DNA. Mutations can have dire consequences since DNA is responsible for holding the information that makes you, you. There's a couple different types of mutations we're going to talk about. The first type is called a deletion. Take a look at this string of children at the top of your screen. What has happened to it in the lower string of children? Well, the one girl child was deleted. It was taken out of the line. That's because now it is a shorter string. This is a deletion. The same can happen to your DNA. Certain letters can be deleted or taken out, which shortens your DNA strand. Take a look at this string of students and the ones at the bottom. They're the same length. However, the girl we have in the top was substituted for a boy on the bottom. This substitution is called a substitution mutation. This is where one is switched out for another. I often compare this to soccer. In soccer, you can make a substitution of one player for another player. It's the same number of people on the field, just you're switching one for a different one. And the last one is an insertion. If you can see, there's now more people in the string of children because we added another girl child. When you add something, it's called an insertion, and it can lengthen your DNA strand. So, let's do an example. At the top of our screen, we have CAT. These are the letters in our normal DNA. What would be our normal mRNA? Well, if you have CAT, it should normally go with G, T, no T on mRNA, U, G, U, A. And you'll have this repeating 3 times. GUA, GUA, GUA. What if there was a mutation? We are going to substitute the letter A for a letter G. What's going to happen to the mRNA now? Is it still going to be GUA? Well, C still goes with G, and T still goes with A, but G does not go with U. G goes with C. So instead, you're going to have G, C, A. This is a different codon, which translates to a different protein. You now have a mutation. How about if we did a deletion? If we deleted the letter T, what would happen? Well, what happens is called a frame shift. You would move the letter T out and shift all the other letters over to fill the gap. This frame shift now means that all of the codons after your deletion have changed. So, none of the codons are the same. CAT is now CAC. That leads to a different mRNA and therefore a different amino acid. This is why deletions are very bad mutations because they cause a lot of damage and a lot of change. The same is true for a substitution. If we were to add the letter T in the front of this DNA chain, there's another frame shift shifting over to make room for this new letter. Now, all of the codons and all of the base pairs after that insertion have changed. That's why insertion and deletion that cause frame shifts are very bad mutations because it causes a lot of change. Now, why would a change be a problem? Well, if you remember, amino acid sequences make proteins. If we're changing the nitrogen bases or those codons, we're changing the amino acids, which means we're changing the protein. Take a look at this normal amino acid sequence. Met, lis, phi, gly, and stop. Stop means we're done reading. This is the resulting protein. What would happen if we were to mutate this? Well, if we were to mutate it, it might not lead to a protein at all, or a completely different protein that our body doesn't need. So, let's review some vocabulary. A gene is a segment of DNA that codes for a specific protein or trait. A mutation is any change in that DNA sequence. A mutagen might be something that caused that mutation. For example, radiation or cigarettes or certain chemicals can actually alter your DNA. A lot of the mutations that happen to our DNA don't cause big problems because a lot of them don't cause frame shifts. However, insertion and deletion definitely cause a frame shift, which means it causes a lot of problems. A gene mutation is a change in the nucleotide that causes a mutated protein. So, here's my normal DNA. Here's my mutated DNA. As you can see, I've substituted the letter A for the letter G. That's not a big change, is it? No, because it's a substitution. A substitution doesn't cause too big of changes, although it still might change the protein at the end. The ones that cause big problems are deletion and insertion. When you delete something, you are removing one of the bases, and that causes everything to shift over to fill in the gap. Take a look here. In the middle example, I have deleted the letter U. What is the resulting protein? Well, as you can see, all of the amino acids after that deletion have been changed. Instead of PHE, we now have LEU. Instead of GLY, we now have ALA. So, and there's not enough bases in order to make a final amino acid. So, let's do some practice on our own. Take a look at the assignments in your Schoology and complete them.